Okay, let's talk about nutrition. So their mode of nutrition, they have a couple of ways that they'll do it. All right, they can be photoautotrophic, which means that they use light as their energy source and CO2 only for their carbon skeleton. These prokaryotes would include cyanobacteria, and then the eukaryotes, which are the descendants of cyanobacteria, would be plants and algae. Chemoautotrophs use CO2 as their carbon source, but they can also oxidize inorganic substances for their energy, which means they're using chemicals for their energy instead of light. This would be all of the hydrogen sulfide bacteria, the ones that will use hydrogen sulfide, ammonia, ferrous ions, and this type of mechanism, chemoautotroph, is only unique to prokaryotes. I mean, there's no eukaryotes, there's no eukaryotic plants or algae that do this. They can be photoheterotrophic, which means that they use light energy to make their ATP. They get carbon from an organic stuff that is, they have to eat, basically, for their carbon. And this is sort of restricted to some prokaryotes can do this, but not all of them. Chemoheterotroph means that they use organic molecules for energy and for their carbon source. All right, most prokaryotes can do this, and basically all eukaryotes can do this. All right, this is how, not can, they do this. So, when we talk about chemoheterotrophs, though, because that's what they like to look at in the book, they're mainly focusing on saprobes, which are decomposers that absorb their nutrients from dead matter, and parasites, which absorb their nutrients from living tissue in a host. Now, the types of nutrients that they need depends on what kind of prokaryote it is. I mean, you know what you've seen how we've kind of screened for some bacteria using different nutrient sources, using different antibiotics. So the necessary growth media varies with each species. For example, there are some bacteria out there that use petroleum as their nutrient source, which is why we've harnessed them to start cleaning up oil spills. If something is non-biodegradable, it means that there is no prokaryote on planet Earth that we know of that will break it down. So that includes some of those synthetic plastics that we've made, which is why it's so important to recycle them. A lot of bacteria, you'll hear about nitrogen fixation. This is unique to prokaryotes alone. We have to have prokaryotes on the Earth to fix nitrogen and fix carbon, because they are able to metabolize most nitrogen-containing components. And this is really, really important in the nitrogen cycle of ecosystems, because this is about the only way that we can recycle nitrogen back into the soil and convert it from dead organic matter back to something that can be used by the plants. Now, nitrogen fixation, just like carbon fixation, means that you're converting like a nitrogen source to another nitrogen source. All right, just like in carbon fixating, when we were talking about photosynthesis, it's converting CO2 into some sort of organic molecule that can be used for energy. When we talk about nitrogen and carbon, they need these things. But one thing that they don't need, unlike eukaryotes, is they don't need oxygen necessarily. So in some cases, you will have obligate anaerobes, obligate aerobes, and facultative anaerobes. Now, them not needing oxygen makes a lot of sense, because back in early Earth there was no oxygen in the atmosphere. So clearly, I mean, they had to adapt some way. So obligate anaerobes, the ones where they live by fermentation or anaerobic respiration alone, oxygen would kill them. They use inorganic molecules to replace oxygen in the electron transport chain when it's going to accept electrons. Those are probably the first types of prokaryotes. And then facultative anaerobes would be, the obligate aerobes would be the ones that, facultative anaerobes would be the ones that can kind of use oxygen if it's present, but can go into fermentation if it's not. And then they, in turn, are the ancestors of obligate aerobes, the ones that are completely dependent on oxygen for respiration and growth. So it depends on what type of, again, it depends on what kind of prokaryote you're talking about, what kind of species. The ones that are the most independent are obviously going to be the ones that are facultative anaerobes, the ones that can do without. You'll see that they live in places that are unheard of for most life. So when we talk about how these photoautotrophs have become the ancestors of plants and algae, we're really talking about cyanobacteria. It's been postulated, and it's probably true, that the first prokaryotes were probably heterotrophic. They got their energy and carbon from organic molecules in the air quote primordial soup of early Earth. Glycolysis, which doesn't require any oxygen or organelles, probably evolved first because it doesn't require any oxygen or organelles. And then natural selection would then favor those prokaryotes that can harness the energy of the sun. So, I mean, these are all things that they postulate. What's the evidence for this? Well, photosynthetic groups of prokaryotes are wide-ranging in all of prokaryotic phylogeny. They hit every single branch of the tree. There's no one place where they're concentrated. Probably means that the photosynthetic machinery probably only evolved once because it's so complex, and then it has been branching out ever since. Now, the second piece of evidence is that cyanobacteria are photosynthetic and very, very old, which means they are probably, the first to evolve. Plus, O2 accumulates at about 2.7 billion years ago, suggesting that cyanobacteria are already here on Earth because then they start to learn to harvest oxygen and sunlight. And we see that there are fossils of prokaryotes that resemble cyanobacteria about 3.5 billion years ago. So, cyanobacteria, with their two photosystems that they have developed, evolved from simpler non-oxygenic photosynthetic prokaryotes. Cyanobacteria changed the atmosphere from a reducing to an oxidizing. Remember that before oxygen was accumulating on the Earth, it was CO2 that ruled the Earth. And with cyanobacteria, they are starting to harness CO2, break it down by carbon fixation, and then as they split water, they're helping accumulate oxygen. And oxygen accumulation drives evolution of cellular respiration, which is common amongst all eukaryotes. So it makes sense that it goes heterotrophic bacteria that didn't use photosynthesis to photosynthetic autotrophic bacteria, and then on to eukaryotes that use respiration and are heterotrophic.